So here's the title of the talk, right? But if you can downgrade it, it's just how diet influences cardiovascular function. Okay? So majority of the work was done at the University of Utah, and I've just moved here and I'm establishing my research lab, and I'm continuing this research. Plus, I'm also kind of expanding into more immune system, immune cells, and we also study aging in our lab, and aging cardiovascular system, aging and inflammation, which is a big topic now because we believe that inflammaging, as we grow older, inflammation sets in, and that promotes aging. So that's the theory behind it, and we are studying a little bit of aging too. So if you look at all these conditions here, healthy aging, not even an unhealthy aging, just healthy aging, physiological aging, diet-induced obesity, insulin resistance, and type 2 diabetes. All these conditions are associated with cardiovascular complications, right? So some person has diabetes and you say like they had a heart attack, and about 70% of people who have diabetes will die from a heart attack, and about 16 to 20% will be dying because of strokes. So those are like large vessel complications basically. You would have taken A&P and you know how vessel, meaning I'm talking about blood vessels, right? So large vessels can result in strokes, atherosclerosis, plaque buildup in your arteries. Those are large vessel complications. And diabetic people, as you can commonly come across, they have this diabetic nerve pain, right? They have issues with their eyes. And then, which is very common, end-stage kidney disease, something that develops with diabetes. Those are all small vessel complications. So why do these conditions lead to cardiovascular complications? Why do they do that? What's the molecular mechanisms? I'm a biology or cell biology person, so the presentation here would be more into the cellular events that take place.
I don't know why it's talking to you like that.
So all these, we are looking at it in a cardiovascular context. If you can tie all these in a cardiovascular context, it all comes to something called as endothelial dysfunction. So what is an endothelium? What are endothelial cells? Do you guys have an idea? What is an endothelial cell?
Oh, come on.
Don't they stimulate vasodilation?
You're right. So we'll go a little bit into the detail and see what an endothelial cell is and how does that influence all this, right? So endothelial dysfunction is the dysfunction of endothelial cells, and that actually is due to a lack of bioavailability of a molecule, a gaseous molecule called as nitric oxide. So nitric oxide is produced in the endothelium, and if you do not have adequate nitric oxide, you develop these kind of complications. So why do we not have, or why do the people who have all these conditions, why don't they have nitric oxide? That's basically what we are going to see today, and how your diet is going to influence that. So if you take an artery, right, your aorta, I'm showing an aorta here. So, and this is a cross section, and you can see the vessel basically, when I say vessel, it's an aorta, an artery. So that is a smooth muscle layer. So you have the vessel that goes like this, right? The outside of it is the smooth muscle layer. So that is the lumen where the blood flows. And if you see here, that is an endothelial cell layer. So you have a single layer of endothelial cells that line the lumen of the arteries. So blood flows through this, and you have a single layer of endothelial cells, and you have smooth muscle cells that surround it. So the endothelial cell is a seat of a lot of activities. It's a hot seat where activities occur that influences the muscle cell that is outside, the muscle layer that's outside. So if you can focus some more, so that's the smooth muscle, that's the endothelium, and you have the endothelial layer of the cell. So what happens is, in the endothelium, you have this molecule called as nitric oxide that is produced. So when nitric oxide is produced, it helps in the relaxation of the muscle outside, or the muscle layer of the artery. So endothelium is the seat of production of nitric oxide, and the nitric oxide diffuses out into the smooth muscles, or the muscular layer, and causes relaxation of the vessels. So as she said, it causes vasorelaxation. The vessels must relax. So for example, if somebody calls you, just come upstairs and take this notebook from me. So you have to get up and walk there, right? So when you get up and walk, there's an increased demand for blood to flow through your arteries. So you get up and you walk, then your blood has to flow into the arteries to help accommodate that. Say your vessels are like that and your blood is flowing and it's not relaxing, there's a pressure there. So as your demand for blood flow increases, your vessels must relax and accommodate that flow. You get up and walk, your vessels relax. Your vessels will only relax if you have this molecule called nitric oxide. This is not available. Usually, you see this in aging. When people get older, this artery, the muscles in the arteries become stiff, and that stiffness is because of the lack of nitric oxide. So nitric oxide is not there, and their muscles are stiff. They get up and walk, or move about. The blood flow is not it's not flowing freely because the arteries are still constricted. That's arterial stiffness. That is a good thing. Good thing in the sense it's a thing that promotes high blood pressure, and people get heart attacks, strokes from that. So the vessel has to relax to accommodate the blood flow. So the primary regulator is nitric oxide, and it's called the master regulator of the entire cardiovascular system. You should have enough nitric oxide. If it's too much, sometimes in certain infections, there are 3 different types of it. A type of nitric oxide increases and is too much, and your vessels just relax, and that time people pass out because of that. So that is an inducible nitric oxide. Usually happens when there's an infection by a bacteria. But we are talking about this constitutive nitric oxide, or the nitric oxide that is constantly present in your endothelial cells. So the endothelial layer is made up of endothelial cells. So that layer has this nitric oxide always, and it's just activated and deactivated depending on the need. Right? It's in a state of activation, but if you have to have an increased demand, then the increase in nitric oxide happens. Right? So now we are going deeper, right? We saw the vessel, we saw the endothelial cell layer. Now if you go and focus on a single cell, so this is a single cell, this is the lumen now, that's where the blood flows, like that. And then you have endothelial cell, and then you have a smooth muscle cell, which should have been bigger like this, but that's a smooth muscle cell. So when the blood flows, when you guys get up and walk, the blood flows, right? When the blood flows, it just drags. The flow just makes a shear stress, or a shear force happens on the endothelial cell. So that shear is actually activating. So the blood flow, your body immediately, or the cells immediately recognizes, oh, the person is walking now, and the blood is flowing, and the cell needs to produce more nitric oxide. What happens? The shear force activates certain receptors here. So receptors are basically, you have a cell, not described very well here. So you have a cell, and the cell has protein receptors sticking out like that. Insulin receptor is something like that, it just sticks out. So these receptors get activated by the shear force, and what happens is they open up and let calcium to come inside. So calcium influx into the endothelial cell happens. So calcium enters the endothelial cell. Now I'm just showing, it's a way more complicated process than what I'm showing here. So I'm just showing the proteins that you need to know about. There are several other proteins involved. So there's another protein called clamodulin. When calcium enters, clamodulin and calcium binds. And nitric oxide, until this one, so it says CNOS, meaning constitutive nitric oxide synthase is the enzyme that produces nitric oxide. So that's an enzyme name. And this is nitric oxide. So constitutive, or you can call endothelial nitric oxide, eNOS. So I'm used to saying it as eNOS, so I'll say eNOS, meaning I'm talking about that enzyme. So this enzyme is a monomer. So single units of it will be just floating around the cell with no purpose, just like that, right? So when shear force happens, calcium enters, calcium clamodulin binding activates them, and they dimerize. They become like two molecules like that. Two monomers stick together and they become a dimer. When they dimerize, the same cell, I'm drawing it bigger here. When they dimerize, they go to the membrane, cell membrane, and stick here. That's called as an area called as caveolae. So they go and hang in there. So when they go there, what has to happen is, so have you read about protein kinases and phosphatases, right? So phosphatases will take away a phosphate group, kinase will add a phosphate group, right? So there is a kinase called AKT, or it's also called as protein kinase B. So that one has the capacity to go and throw a phosphate group on the enzyme eNOS. So proteins have a complicated structure, and each site is given a name, like serine 1177. So that's the site that is phosphorylated, the site on the protein that takes a phosphate group from AKT. So that phosphorylation happens. So you have another protein that sticks to, that's called AKT. So all these would happen as soon as the shear happens, calcium enters in, AKT sits with eNOS. Right, so far clear? You guys are okay? So AKT sits with eNOS, puts a phosphate group on. Now something has to keep this eNOS and AKT together. So there's another protein called as HSP90. That's like, so you have eNOS, then AKT, and HSP90 is like a clamp that keeps them together. So it's an adapter protein. So eNOS, AKT kept together by HSP90. This kind of interaction happens. When this happens, eNOS becomes active. Now, eNOS is an active enzyme. After all these things, the active enzyme, you have a substrate called L-arginine, an amino acid in the cell. The enzyme-substrate interaction, enzyme breaks down the substrate. The product is you have nitric oxide and citrulline. Let's forget about citrulline. Let's focus on nitric oxide. Now, nitric oxide is produced by the activated enzyme. Nitric oxide diffuses into the smooth muscle layer. So you had like that single layer of endothelium, it diffuses into the smooth muscle. So when nitric oxide enters the smooth muscle, then once again a lot of protein interaction. It activates an enzyme called as guanylate cyclase. So now guanylate cyclase actually shuts down calcium receptors on the smooth muscles. So when calcium is shut down, what happens is smooth muscle relaxation occurs. Calcium in its opposite reaction here. So calcium enters, activates eNOS in the endothelial cell. Once nitric oxide diffuses into the smooth muscle cell, the activation actually shuts calcium from entering into the smooth muscle. That causes the relaxation. So this is the process that happens when somebody calls you upstairs. So you get up and run there, and all these kind of things happen, right? So now your vessel relaxes, and the blood continues to flow freely. So that has to happen. If it doesn't relax, then it's just so tight, and the blood is flowing with a pressure inside. That kind of pressure, constantly, a pressure flow of blood in actually damages the endothelial layer. The damage, when your body senses a damage, is something called as inflammation that sets in. All kinds of tissue damage starts to happen if that relaxation doesn't happen. So you need that relaxation for normal function. So apart from being a vasorelaxant, nitric oxide can be called as a vasorelaxant. Apart from that, it has several functions. This slide is a bit complicated, you don't have to think of it too much. So first is, One is it's a vasorelaxant. Second is in the lumen you have blood platelets, cells of the blood. They sometimes can stick together and then deposit inside the artery and form the plaque buildup, right? Nitric oxide can prevent that. So nitric oxide, free flow of nitric oxide actually prevents inflammation, prevents platelet from aggregating. It's an antioxidant too. So when your cells metabolize, they produce superoxide. Superoxide is basically an anion of oxygen and it has one electron less, so that superoxide can combine with hydrogen and form hydrogen peroxide, and it's a molecule that signals in the cell. However, if it's too much, it becomes toxic to the cell. Nitric oxide can quench that by activating some enzymes, and it's got the capacity, it's an antioxidant. So two benefits of it. Another thing is it maintains the vascular tone, making it relax, contract, so vascular tone is maintained. Homeostasis, keeping everything in order, like the steady state. That function is also nitric oxide. The next one is vascular permeability. So the nitric oxide keeps the vasculature intact. If your endothelium becomes leaky, then there's a damaged aorta, right? So you want it to be intact. So vascular permeability is maintained by nitric. So it's got all these benefits. So you do need nitric oxide at adequate quantity or appropriate quantity to maintain your vascular function. So what happens? So this is basically what we know about nitric oxide, right? So why does nitric oxide reduce? Or why does its bioavailability, why does it go down with conditions that we mentioned earlier? So one condition that we studied was a high-fat diet, right? High-fat, we studied it as a diet-induced obesity. So obesity comes under that, right? Obesity, diabetes, all these conditions somehow promote or dysregulate nitric oxide production. How? So if you have a food that is high in saturated fatty acids, for example. Your food, when it's digested, ultimately if you have a fatty food, the fat is broken down into fatty acids, right? Free fatty acids. If you had a sugary or a carbohydrate-rich food, the final units are glucose molecules, and then glucose units. Then if you had a protein, it gets broken down into amino acids, and then you use it for building tissues. All these things happen. Or energy, ATP production goes into the cell, glycolysis happens, and then you go into the mitochondria, ATP production. So you use it up like that. What happens is if you have a fatty diet, you get these small free fatty acid molecules. If you have excess of free fatty acid molecules, something happens to nitric oxide. It results in a decrease in eNOS activation, right? Decrease in eNOS activation. That results in decreased NO production. So if you have decreased NO production, what happens, as I mentioned earlier, plaque buildup occurs. Nitric oxide is not there to keep check of certain platelet aggregating factors. So platelet starts to stick, and your endothelium also upregulates certain molecules called as vascular cellular adhesion molecules, meaning they are like gums, sticky material comprising of cells and proteins that are all upregulated on the endothelium, and which causes the cells that flow in your blood to stick, especially a cell type called monocytes. They just stick there and cause a foam cell. So you see how plaque builds up. Nitric oxide bioavailability goes down. Somehow fatty acids do that. We are going to see how they do that. And that results in this plaque buildup.
No, this is really good. I just had a question because
Sure.
it's fascinating. So you're probably going to cover it in a slide or two, but with the free fatty acids in this interaction, is that, I assume that it is modified by the type of fat being consumed?
Sure,
So,
definitely.
so you'll touch on that? Okay.
So in our lab, so we are just using palmitate as a surrogate
Okay,
for
yeah.
the fat, and then oleate, which is an unsaturated fatty acid, and
Okay.
we do see a lot of them has been seeing a little bit of it. So we are seeing the differences in the type of fat that you eat definitely influences. Okay? So in this case, I'm
Cool.
talking about free fatty acids, and that is from saturated fats. So you see how plaque buildup occurs over time. See how that builds up and how your artery is so narrow. And then obviously your blood flow will be so restricted in that, right?
Did you turn it off? No, I think it probably just went to sleep. It does that to me every once in a while too. Just the computer, it's not the. Oh, it's this button. All that matters is we get it back, right? You have to finish the story though, because I'm waiting for the.
Okay. So now you see what happens with the lack of nitric oxide, and how does this free fatty acids do this? You know, obviously there are like several mechanisms, and we were focusing on free fatty acids, right, from saturated fats. So how do we study this, right? Best way is the 4 mouse. So we had a mouse model.
It's always the mice.
It's always the mice. So we have a mouse model. So what we did, we took 2 groups of mice, one on a high-fat diet. So the diet was 45% from saturated fats, 35% from carbs, and then 20% from protein. Right? Then a controlled diet, which is 10% from fat, 10% of energy or energy from fat, and then 70% carbs, and then 20% amino acids. So that was the diet composition. So what we did, this is basically characterization of the model. So you start doing something, you want to see the effects, right? So I'm just testing out. So we put the mice on a high-fat diet. Do they gain weight? Yes. High-fat fed mouse gained weight. They were fatter than the low-fat mouse. And then
Poor mice.
with high-fat diet, we started seeing impaired glucose tolerance. You guys know glucose tolerance tests? You guys done that. So in mice, what we do, we just take the mouse and then inject it with glucose at different time periods. So what happens is, we have a slide, okay, here's the slide. Okay. So when you have a glucose high, you have a food or a meal, glucose enters your system, that makes pancreatic beta cells to produce a hormone called as insulin. So the insulin will bind to receptors, I said those receptors, insulin will bind to receptors on cells. So when it binds on the cell receptor, what happens, several signal transduction, protein-protein interaction occur that will result in the opening up of transporters of glucose called as GLUT1. You have GLUT1, GLUT4, and all kinds of transporters. So those are transporters, meaning they transport glucose from the blood into the cell. So if you eat a meal, normally everything happens, insulin is produced, binds, the GLUT receptors will open up, take up the glucose into the cell, chop it up, glycolysis, mitochondrial electron transport chain, and then you have ATP. If that doesn't happen, so if your cell becomes resistant, insulin is there, your cell is not responding to that insulin, that's where you start getting insulin resistant. Your insulin is here, but your receptors are not even responding, just pretending as though insulin is not even there.
La 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 la.
It's just closing its eye and just staying there. If that happens, the glucose transporters will not open up. The glucose in the blood will not enter the cells, and the glucose lingers in your blood. That's why over time you see blood glucose, high blood glucose. That's what happens, right? So these mice, so that's how we test. So you give them a shot of glucose, wait for some time for them to clear it, and then you measure their blood glucose. So your blood glucose, so this is area under the curve. So if it's a controlled mice, you give them a glucose shot, they clear it off very fast, they clear it off soon. But a high-fat mouse, it takes a longer time for the area under the curve is plotted here. So you take a longer time for the high-fat fed mouse to clear the glucose, meaning it's lingering there. So it's becoming insulin resistant, and its glucose tolerance test is not right, because you see glucose not being cleared. So yes, that happens with a high-fat diet. So the mice are becoming resistant to insulin. They have glucose in their blood. Then you're feeding them a high-fat diet, so you should see more of free fatty acids in their body. So we measure the free fatty acids. Control-fed mice, less compared to that. The high-fat fed mouse, you had like significantly high levels of free fatty acids. Free fatty acid, mostly from our diet, we get something called as palmitate, which is C16. You know, the fatty acid have like 16, 12, 13, all this kind of thing. So 12, 14, 16. So C16 is a common type of fatty acid that's found in our, if we have a high fat diet. So the mice are becoming like how we want them to be.
Like
The
Americans,
model-
they're becoming like Americans.
The model is working, right? So that's what we do when you set up an experiment. You want to see if your model is working. So the model is working here. This I explained earlier. So now we want to see ENOS function. So this is, have you guys seen a Western blotting? You guys done some Western blotting? Some, yeah. Okay, this is a technique. I'm not going to go into detail. So how you do that is, say in this case, we fed the mouse, right, with a high-fat diet. We are interested in arterial ENOS. ENOS is found in that endothelial cell, right? So what we do, we chop the artery, a piece of it, grind it down, grind the tissue, make the protein release. So chop the tissue, get the proteins released out. Now take the proteins and run them out on a gel. Like your DNA gel, this is a protein gel. So you run it on a gel. So as I mentioned here, more phosphate group added to ENOS. This is not always the case, because in certain cases when phosphate group is removed, the protein can become activated. In ENOS, you're adding phosphate group to that site serine 1177. So if you have a thicker band there, it means it's got more phosphorylation. So the technique I'm not going into, but just understand, if you have a thick band there, it means more phosphorylation and ENOS is active and more nitric oxide is produced. It's a indirect way of testing if you have enough of nitric oxide. We can test nitric oxide itself. We have done that too. But this is a common technique. Nitric oxide assay as such is very time sensitive. You have to be so quick to go and measure it, so it's a little complicated. So we just go with a Western blotting and see how the protein is phosphorylated. So that's an easy way for us to do. So we looked at basal and after insulin stimulation, because if you give a stimulus, for example, I mentioned you get up and walk, that's when you have increased blood flow. If you're sitting, your blood flow is just normal. And you have a basal level of ENOS activation, that's it. You give an agonist a push, then you get more phosphorylation. How do you make a mouse, like how do you give a push to the mouse? It's running all the time. So the best way to stimulate ENOS, it has its basal level. And then if you want to stimulate it, you just give it a shot of insulin. So if you look at a controlled diet fed mouse, basal and after insulin is an agonist, insulin is an agonist, you see a robust phosphorylation of ENOS. But in a controlled mouse or in a high fat fed mouse, you see ENOS phosphorylation is so less, meaning the nitric oxide production is also less here. So high fat diet not only influence the free fatty acid level, blood glucose level, and the weight gain, it also influences ENOS levels. Okay, so how does it do? That's the mechanistic one we're going to get into. So you saw ENOS activation, you saw the mouse are like heavy and they're having issues. Now, how is your artery functioning? Is it relaxing and contracting? We know that we have less of ENOS here. What's the functional readout? Meaning, if you have less of ENOS, your vessel should not relax. It should be tight, right? So next measure is done by something called as a myograph. So you take the artery of the mice and then you inject it with, like insulin, acetylcholine is an agonist. If you inject acetylcholine, the vessel should relax like that. So here we have different doses, on the x-axis we have different doses of acetylcholine, and this is relaxation, percent relaxation. You hit it with the acetylcholine, it has to relax. You hit it more, it has to relax more. So that has to happen here. If you see that control